chitosan zeolite composite materials for the removal of heavy metals and the inhibition of Escherichia coli in wastewater. Introduction. Water contamination by heavy metals and pathogenic microorganisms is one of the most relevant environmental and public health problems worldwide due to its direct impact on ecosystems and human health. Industrial growth, accelerated urbanization, and inadequate waste disposal have significantly increased the presence of chemical and biological contaminants in surface and groundwater. Among the most hazardous contaminants are heavy metals, particularly lead and cadmium, owing to their high toxicity, environmental persistence, and bioaccumulation potential. These metals are non-biodegradable and can persist in the environment for long periods, causing neurological, renal, cardiovascular, and skeletal damage, as well as potentially carcinogenic effects. In parallel, microbiological water contamination represents a constant threat to public health. The presence of Escherichia coli is widely used as an indicator of fecal contamination and the potential presence of pathogenic microorganisms capable of causing gastrointestinal and infectious diseases. Consumption of or contact with contaminated water can lead to outbreaks of disease, particularly in regions with limited access to efficient water treatment systems. Therefore, there is a growing need to develop technologies capable of simultaneously removing chemical and biological contaminants in an efficient and sustainable manner. In this context, the synthesis of materials capable of removing contaminants from polluted water has been explored as an efficient and operationally simple alternative to conventional treatment methods. In recent years, composite materials prepared from biopolymers and natural minerals have attracted considerable scientific interest because they can combine structural, chemical, and biological properties for the simultaneous removal of different contaminants. Chitosan zeolite composite materials for the removal of heavy metals and the inhibition of Escherichia coli in wastewater. Introduction. Water contamination by heavy metals and pathogenic microorganisms is one of the most relevant environmental and public health problems worldwide due to its direct impact on ecosystems and human health. Industrial growth, accelerated urbanization, and inadequate waste disposal have significantly increased the presence of chemical and biological contaminants in surface and groundwater. Among the most hazardous contaminants are heavy metals, particularly lead and cadmium, owing to their high toxicity, environmental persistence, and bioaccumulation potential. These metals are non-biodegradable and can persist in the environment for long periods, causing neurological, renal, cardiovascular, and skeletal damage, as well as potentially carcinogenic effects. In parallel, microbiological water contamination represents a constant threat to public health. The presence of Escherichia coli is widely used as an indicator of fecal contamination and the potential presence of pathogenic microorganisms capable of causing gastrointestinal and infectious diseases. Consumption of or contact with contaminated water can lead to outbreaks of disease, particularly in regions with limited access to efficient water treatment systems. Therefore, there is a growing need to develop technologies capable of simultaneously removing chemical and biological contaminants in an efficient and sustainable manner. In this context, the synthesis of materials capable of removing contaminants from polluted water has been explored as an efficient and operationally simple alternative to conventional treatment methods. In recent years, composite materials prepared from biopolymers and natural minerals have attracted considerable scientific interest because they can combine structural, chemical, and biological properties for the simultaneous removal of different contaminants. Methodology. The chitosan and zeolite-based composite was synthesized using the ionic cross-linking method. The clinoptilolite-type zeolite was ground and sieved to obtain a particle size of 200 micrometers. It was then washed with deionized water and dried in an oven at 110 degrees Celsius for 24 hours. Next, 4.5 grams of zeolite were dissolved in 20 milliliters of deionized water. A 3% chitosan solution was prepared by dissolving 1.5 grams of chitosan in 50 milliliters of 5% acetic acid. Both solutions were mixed for 4 hours. After stirring was complete, the chitosan zeolite mixture was added dropwise to a cross-linking solution of sodium tripolyphosphate. Subsequently, the composite was washed with distilled water until a neutral pH was reached and dried at 35 degrees Celsius for 72 hours. The antimicrobial activity of the composites was evaluated using Escherichia coli strain ATCC 25922. Standardized bacterial suspensions were prepared, described the inoculum preparation procedure, and adjusted to a turbidity equivalent to 0.5 on the Macfarlane scale. Subsequently, 0.5 milliliters of the bacterial suspension was inoculated into tubes containing 10 milliliters of soy tryptocasium broth, establishing the following experimental groups: C, CZ2%, CQ2%, QZ-PB0.25%, and QZ-TPP2%. The cultures were incubated at 37 degrees Celsius for 24 hours under aerobic conditions. At the end of the incubation period, bacterial growth was assessed by measuring the absorbance at 625 nanometers. Subsequently, each culture was subjected to a serial dilution from 1 to 1000, and from this, an aliquot of 0.001 micrometers was inoculated using a calibrated loop onto tryptocasian soy agar using the surface streaking technique. The plates were incubated again at 37 degrees Celsius for 24 hours under aerobic conditions, and colony-forming units were subsequently counted. Data were expressed as mean plus or minus standard deviation, or as median and interquartile range, depending on the data distribution. Normality was assessed using the Shapiro-Wilk test. To compare turbidity and colony-forming units among the experimental groups, a one-way analysis of variance was performed, followed by Tukey's post-hoc test when the assumptions of normality and homogeneity of variances were met. Otherwise, the non-parametric Kruskal-Wallis test was used, followed by the Dunbon-Ferroni correction for multiple comparisons. The lead and cadmium removal tests were conducted using solutions containing 200 milligrams per liter of each contaminant. 40 milliliters of solution were placed in 50 milliliter polypropylene tubes, and 0.1 grams of composite were added. The samples were kept in contact for 7 days at 25 degrees Celsius, with the pH adjusted, lead to 5, cadmium to 7, using 0.1 N sodium hydroxide and hydrochloric acid solutions. Results: antimicrobial activity. The results indicate that the antimicrobial activity of the evaluated composites is primarily determined by the presence of chitosan. The 2% composite showed inhibition of E. coli growth comparable to that observed in the chitosan control, both in terms of the decrease in absorbance and the reduction in the number of CFU, demonstrating that its incorporation into the clinoptilolite matrix and subsequent cross-linking with sodium tripolyphosphate did not significantly affect its antimicrobial properties. The bactericidal activity of chitosan has been extensively documented and is attributed to the protonation of its amino groups in slightly acidic media, which confers a positive charge capable of electrostatically interacting with the lipopolysaccharides and phospholipids of the outer membrane of gram-negative bacteria. This interaction alters the permeability of the cell membrane, promotes the loss of intracellular components, and inhibits bacterial growth. Furthermore, the antimicrobial efficacy of chitosan depends on factors such as its concentration, molecular weight, and degree of deacetylation, which determine the intensity of the interaction between the polymer and the bacterial surface. The lower activity observed for 0.25% QZ-PP confirms that the antimicrobial effect depends on the concentration of chitosan present in the material. A higher proportion of the biopolymer increases the availability of protonated amino groups and promotes contact between the composite surface and bacterial cells, thereby enhancing cell membrane disruption. This behavior is consistent with what has been reported for chitosan nanoparticles and cross-linked materials, in which an increase in polymer concentration results in greater antimicrobial activity. These results also show that clinoptilolite, used as a control, did not exhibit a noticeable inhibitory effect against E. coli, as evidenced by the high absorbance values and CFU counts. These results confirm that the antimicrobial activity of the composite is associated with chitosan and not with mineral support. In this context, the primary function of zeolite within the composite is to provide structural stability and act as an adsorbent matrix, while chitosan is the bioactive component responsible for bacterial inhibition. The structural stability of the composite is closely related to cross-linking with sodium tripolyphosphate, since the formation of ionic bonds between the amino groups of chitosan and the phosphate groups of the cross-linking agent creates a more stable structure in an aqueous medium, reducing the polymer's solubility without compromising its antimicrobial activity. The preservation of activity observed for the 2% QZ-TPP composite supports this behavior. Several studies have shown that ionic gelation with TPP improves the physicochemical stability of chitosan-based materials and allows them to retain their bactericidal capacity. Lead and cadmium removal. The maximum adsorption capacities for lead were 77.17 milligrams per gram at pH 5, while for cadmium, values of 30.07 milligrams per gram were recorded at pH 7. The pH values studied were selected based on the aqueous speciation of both metals, avoiding precipitation conditions, in order to ensure the presence of the cations in solution. Characterization. Microscopic images of the composite reveal an irregular and rough surface, attributed to the incorporation of zeolite into the polymer matrix. The ratio used facilitated the encapsulation of the zeolite structure, leading to the formation of the composite. In the infrared spectrum of chitosan, the functional groups characteristic of this biopolymer is observed. Hydroxyl at 3,358, carbon-hydrogen bonds at 2872, amino at 1580 cm superscript 1, and ethers at 1025. In zeolite, a band associated with hydroxyl group vibrations is observed at 3,600, one at 1627 attributed to the aluminosilicate structure, as well as the typical silicate vibrations at 1,015. The spectrum corresponding to the composite shows synergy between its components. The curves showing the percentage of weight loss and the rate of weight loss indicate that the materials exhibited total weight losses of 64.68% for chitosan, 11.11% for zeolite, and 32.3% for the composite. In all cases, an initial loss was observed below 100 degrees Celsius, attributed to water evaporation. Chitosan exhibited a marked loss at 256 degrees Celsius, associated with its thermal and oxidative decomposition, as well as with the vaporization and release of volatile products. In contrast, the zeolite showed only a loss of residual moisture in its pores, confirming its high thermal stability up to temperatures below 600 degrees Celsius. The composite, meanwhile, exhibited weight losses corresponding to both moisture evaporation and the degradation of the organic phase of the chitosan. Conclusions. In this study, we successfully synthesized a chitosan zeolite composite cross-linked with sodium tripolyphosphate, yielding a material with structural and functional properties suitable for potential application in the treatment of contaminated water. Evaluation of its antimicrobial activity against Escherichia coli demonstrated its potential as a multifunctional material for the comprehensive treatment of chemical and biological contaminants in aqueous systems. The results showed that the 2% SB3-P046 composite significantly reduced both turbidity and Escherichia coli colony-forming units compared to the control groups, confirming an antimicrobial effect attributed primarily to the protonated amino groups of chitosan. Furthermore, it is important to note that clinoptilolite zeolite alone did not exhibit antimicrobial activity. However, within the composite, it acted as a structural support, while cross-linking with sodium tripolyphosphate promoted greater stability of the system in an aqueous medium. The results obtained demonstrate that the composites have high potential for the removal of lead and cadmium, with maximum adsorption capacities of 77.1 and 30.07 milligrams per gram, respectively. Infrared spectroscopy analysis confirmed the presence of functional groups characteristic of chitosan and the structural bands of zeolite. Thermogravimetric analysis revealed that SB3-P046 exhibits greater thermal stability than pure chitosan due to its interaction with the zeolite and tripolyphosphate. Overall, the composite represents a promising, sustainable, and environmentally friendly alternative for the development of water treatment technologies capable of simultaneously removing chemical and biological contaminants.